Welcome, everyone. We're going to get started. So welcome everyone here in the room, and apparently lots of people on Zoom to the first INN seminar of the school year. Big welcome to all of the new students. Okay, so we're going to start it off with a bang today with a seminar from Alex Wiesman, who is, we were just saying, still surprisingly to me, a pretty new system professor. So he just joined us last summer in the Department of BPK. And he comes to us after completing a PhD at the University of Nebraska Medical Center and a postdoc as a CHR Banting Fellow at the Montreal Neurological Institute at McGill University. He's had an impressive start as an assistant professor. So already holds a Canada Research Chair, Tier 2 in Neurophysiology of Aging and Neurodegeneration. And he's the scientific director of our ImageTech lab at Surrey campus. So a really busy guy. His research uses neuroimaging to study neuromodulatory systems in aging and neurodegenerative diseases like Parkinson's and Alzheimer's disease. And I'm sure you'll see a lot about that type of research today. Alex is also the inaugural chair of the INN's training committee, which is full force underway now, but is there still room for new trainees to join? I would assume if they're interested.
We're always looking for input, and yeah, we're happy to.
So keep that in mind if you're interested in that. And we're all really grateful and lucky to have Alex's leadership here in pushing forward new training initiatives at SFU and INN. So everybody help me welcome Alex. Really looking forward to hearing about your research.
Great. Thanks so much for the intro, Lisa. Thanks everyone for showing up here and for the folks on Zoom. I'm excited to be here, talk a bit about the work that I've been doing and that my lab's continuing here at SFU that uses functional brain imaging, so measures of how the brain's signaling, to try to understand why some people develop age-related neurological disease, and probably equally as importantly, why people differ in how they sort of express these diseases, how their symptoms develop and how they develop different profiles of symptoms. So we think this is a really important question to answer because, as probably a lot of people here know, the world population is aging. By that, I don't mean that everyone sitting here listening to me talk is just getting older minute by minute. That's definitely true. You're probably getting much older because I'm the one talking. But what I mean by that is actually that the percentage or the proportion of people in the world who are 65 years of age or older is getting proportionally larger year by year. There's lots of ways to represent this, but in a recent report from the UN that you can see on the left here, you can see that over the next 30 years or so, it's expected that the proportion of people who are 65 or older is going to double, and within the next 60 to 70 years, it's supposed to at least triple. So there's going to be a lot more folks who are above 65 years old, and unfortunately, that brings with it a higher incidence of age-related neurological disease. So diseases like Alzheimer's and Parkinson's disease are diseases primarily of aging. We oftentimes think of risk factors for these diseases in terms of modifiable risk. That's for obvious reasons, right? We research things we think we can help people to change to try to avoid these diseases. But outside of these modifiable risk factors, if you kind of take a holistic look and step back and look at what's actually predisposing people to these diseases, by far the most predictive factor is chronological age, how long you've been on Earth. So being above 65 years of age is by far the greatest risk factor for developing Alzheimer's and Parkinson's. So since so many people are going to develop these diseases in the near future, we need to get better at understanding who's going to develop these age-related neurological problems and why. So we need a better toolkit for understanding this. And unfortunately, that's a really difficult nut to crack. And that's because from recent research using positron emission tomography, for instance, that's able to measure the harmful accumulation of proteins in the brains of patients with Alzheimer's, as well as parallel lines of research in Parkinson's. We know that these age-related neurological diseases have a very long preclinical silent period. What I mean by that is that there's pathological accumulation of these proteins and other pathological processes that happen in the body and in the brain, oftentimes for decades before people show up to clinic with memory problems in Alzheimer's, with movement problems in Parkinson's. So we really need to be in this early preclinical stage trying to target these pathological problems that are starting to occur early on before we know that there's actually an issue. So we need a strong toolkit of measures that we can use to actually understand when these pathological processes are starting in healthy older adults, or at least at the time healthy older adults, And unfortunately, the measures that we have now are largely non-repeatable and relatively invasive. So things like amyloid PET that we're showing here on the graph, that I think everyone's probably seen ad nauseum at this point, is relatively invasive, right? We expose people to ionizing radiation, we inject them with radio tracers, so it's not particularly repeatable, especially if we're talking about using it as a screening tool or as a predictive tool along with other risk factors. So we really need better non-invasive and repeatable measures of risk for pathological aging trajectories, and we need them as soon as possible. One potential measure that we could foresee being a part of this really, really essential toolkit is functional brain imaging. So by mapping the brain's signaling alongside other risk factors, our group and others has been seeing that we may be able to better predict who's going to start developing these pathological trends in brain function. And the method that my group uses primarily is called magnetoencephalography, or MEG. This is a whole brain method, so it's really nice. We get really good spatial resolution measures across the entire brain of how the brain is signaling while someone's performing a task like you see in the image here, or oftentimes as someone's just resting quietly in the scanner. All of the data I'll be talking about today is resting state or task-free, but of course really interested in the task-based stuff as well. Just can't fit it all into one talk. The other piece of MEG that we really like, probably even better than the spatial resolution, is the temporal resolution. So with MEG, we sample at least once every millisecond, if not faster. And that's really essential because with this type of brain imaging, we're able to get these different speeds of brain signaling out of our data, right? So for every single location in the brain, we can decompose a continuous signal that looks something like this, that's kind of messy looking, into rhythmic and arrhythmic components that are different speeds. And it turns out, and I'll talk about this a bit on the next slide, it turns out these fast patterns of brain signaling that aren't resolvable with other methods are actually really, really important for understanding pathology in age-related neurological declines. So MEG has really high temporal spatial accuracy, covered that. The spatial part will be really important for the talk today, so keep that in mind as well. It's also non-invasive, very low risk, and extremely repeatable. We oftentimes joke that the biggest risk of MEG is being extremely bored because you end up sitting in a chair for an hour and staring at a screen, right? That's not very fun, but it is a completely passive measurement technology. It's also accessible widely to lots of different patient populations. It's quiet, so oftentimes it's useful in a lot of patient populations. For instance, folks with mild cognitive impairment or dementia who may not be amenable to other research scanning environments. And as I mentioned before, one of the things we really like about this is the ability to resolve those different speeds of brain activity, particularly really fast patterns of brain signaling. And in previous research that I've been a part of, as well as from other groups, it's been found that these fast patterns of brain signaling are really what's useful for tracking pathology in these age-related neurological declines. So in previous research, we've shown that these fast patterns of signaling track really sensitively the accumulation of amyloid proteins across the brain, as well as tau proteins. These are the two proteins that are known to be sort of the harmful pathological earliest, or some of the earliest features of Alzheimer's disease. And we've also shown that this extends into the preclinical asymptomatic phase. So not only can we look at, can we kind of predict where amyloid's accumulating in the brain from these patterns of brain signaling changes, we can also actually see, based on that reactivity to these pathological accumulations, we can actually predict who's going to eventually start declining cognitively in the long term. We see similar sensitivity in Parkinson's disease. We also see that these fast patterns of brain signaling are really sensitive measures of who's developing more of a cognitive type phenotype in Parkinson's disease, versus who's actually also just sort of developing the more classical motor type profile. So I'm not going into these studies in detail right now, but basically this is just background to say that these rhythmic fast patterns of brain signaling seem to be really important for understanding pathology in these age-related disorders, even using these more sort of classical neuroimaging approaches where we're just testing one thing against another, doing statistical thresholding, and then waving our hands a bit after the fact. So we've found these interesting relationships between brain rhythms and pathology in age-related disease. But more recently, we've been taking a step back in my group and in groups that I've been working with, and asking how should we actually be studying these patterns of brain change? Again, everything that's been done traditionally kind of looks like what I showed on that last slide, where we have one brain map for each of our participants, right? These can be patients or healthy controls. Usually it's a mix of both, and we contrast that itself. But alongside these brain maps, where we have, let's say, rhythmic signaling in each region of the cortex for each of our participants, we also have a feature of interest, right? A contrast of interest. This could be a group contrast. It could be patients versus healthy controls. So we're looking for general effects of the disease process. Or it could be some measure, a continuous measure of disease severity, for instance. So this could be something like the Montreal Cognitive Assessment in Alzheimer's, where we're looking for who's having worse cognitive symptoms and relating that back to the brain signaling features that we're interested in. So what we normally do in sort of our classical approach is we take this contrast of interest, we take our brain imaging data, and for each region across that brain, we model things, right? We do a linear model. Sometimes we feel fancy and we do a nonlinear model. But basically, for each region of the brain, we're fitting a model between our external feature of interest, our brain data, and then we get this statistical map, right? That gives a statistical effect per region of the brain and shows oftentimes a pretty interesting spatial topography, right? A spatial pattern across the map. But the next step is really what we've been questioning recently in the field, which is that we typically take the inferential statistics associated with these effects, right? Things like p-values, and we somewhat arbitrarily threshold that brain map, usually controlling very stringently for multiple comparisons across several brain regions. And we focus in only on the brain regions that are showing what we think of as significant effects, right? So we'll end up having 2 brain regions maybe out of this entire map of hundreds of regions, where we say, this is the brain region or the set of brain regions that are responsible for brain activity changes in Parkinson's, right? Or this is what's related to cognitive declines in Alzheimer's disease. And then, of course, to contextualize those findings, wave our hands around wildly and say those regions tend to be rich in acetylcholine, so that's why it's important. Or those regions tend to be important in other studies for attention, for memory. And we do a lot of what's called reverse inference, right? We reverse infer what those regions are important for in other studies, so they must be important here. That's not a great practice, and it's not very scientifically stringent, right? And implicitly, what we're assuming when we do this, when we focus in only on those very narrowly defined and very overcorrected brain regions, is we're assuming focality in how the brain processes information. Inherently, we're saying these brain regions are what's important for Alzheimer's. These brain regions are what's important for cognitive declines in Alzheimer's disease, which in some cases is useful if we're trying to, for instance, find new spatial targets for a non-invasive therapy. Maybe we do want to target the regions that are most involved in this process. But based on what we know in modern neuroscience about how the brain functions, we know that focality is really not where it's at. It's actually distributed processing across brain regions that's important for all of the complex cognitive and behavioral things that we like to do in the world and we want to do, and that are impaired by age-related diseases like Alzheimer's and Parkinson's disease. So there's been this shift in the field towards kind of straying away from this overcorrection of maps at this initial statistical step, and instead looking at the spatial pattern of what's occurring across the entire brain, and then doing contextualization or looking at things like associations to neurochemistry, to cognitive function, using that entire map. So this is a really nice recent opinion paper that came out from Bratislav Misik's group, where they basically argue that when you over-threshold these maps, you highlight regions, like region-wise associations, but as you progressively unthreshold these maps, you find what's probably a lot more interesting, which are these spatial topographic patterns, right? That span the entire brain. And then plus an assumption here is that the region where we actually don't find associations, that's meaningful too, right? It's meaningful that we find associations in some regions and not in others. And again, this really ties into the idea that we know now that the brain does not coordinate behavior, does not coordinate cognition, and does not likely, in most cases, find changes that are responsible for an entire disease process in single focal regions or very small sets of regions. So what we can do instead is use what's called topographical analysis. We can take whole brain maps of these sort of spatial topographies of what's happening across the entire brain, and we can consider those alongside some of these new fancy new tools that we have that I'll talk about in a second, to consider brain changes across the brain rather than focusing in on single brain regions and then trying to reverse infer what's happening after the fact. I do want to say that this is not a new idea. There's kind of a running joke in cognitive neuroscience in the field that if you think of a cool idea, Randy probably thought of it 10 years ago, and that's definitely true here. Randy and some co-authors in '96 were using functional PET to study memory. And he developed this really cool idea where you can actually use what's called partial least squares, which is a really interesting statistical approach. I'm not going to go into it in too much detail today, but happy to talk about it after if folks are interested. But basically, you can use this new statistical approach to, instead of finding single regions and doing region-wise modeling and seeing where in the brain, in this case, memory is being coordinated in a single location, you can actually look for the spatial patterns across people that seem to be coordinating memory across the entire brain at once, and then interpret those instead, which is kind of the precursor to what we're talking about today. So topographic analysis is not new at all, but what is new and what's really reinvigorated this idea in the field is that we now have these really fantastic and comprehensive resources that help us contextualize these spatial topographies in a much more meaningful way. We have things like the Allen Human Brain Atlas, which is a whole brain atlas of spatial transcriptomics. So for most genes you're interested in, you can have this spatial map of where those genes are most strongly transcribed versus other regions, again, covering the entire cortex. We also, and this will be the focus of the talk today, have this really fantastic resource of neurochemical atlases. We have a multi-atlas of where in the brain we expect higher densities of certain neurochemical systems versus others. And even more recently, we have really more niche but really exciting atlases of things like mitochondrial respiratory capacity, right? So we have, through some really terrifyingly difficult legwork by, I'm sure, some poor graduate student, this really fantastic atlas of where in the brain we see reliance on a really high respiratory capacity by mitochondria. So focusing in on the neurochemical aspect of this, my group uses all of these atlases in different ways and for different use cases. But in this case, we're talking about neurochemistry. So the NeuroMaps multi-atlas, some people here might have heard of it, is this really fantastic resource that was developed a few years ago. I think Justine actually gave one of these seminars a couple of years back. So this is piggybacking off of her and Bratislav and Ross's efforts. But this atlas was developed in Montreal, actually, while I was there doing my postdoc. And basically what Bratislav's group did was cold call a bunch of labs around the world. They called dozens of labs that specialize in positron emission tomography or PET research all over the world. And these groups specialize oftentimes in one specific PET tracer. So they're able to look, there's one group that specializes in measuring norepinephrine transporter, and that's really their whole bread and butter, and they're the best at it. And they cold called all of these groups. They said, hey, listen, we understand you probably can't give us, for ethical reasons, individual data of these PET tracers across the brain, but maybe you could share with us an average map instead. There's no identifying information in there. There's no ethical concerns there. And they got a really strong uptake. They got about, I think, 1,200 plus individual PET maps that ended up being contributed to their database. And what they ended up with was this multi-atlas of neurochemical system density across the healthy human cortex, covering all of the neurochemical systems that you see here. So we're hitting a lot of the ones we're interested, especially in age-related disease. Things like serotonin, acetylcholine, norepinephrine, and dopamine are all included here. And we can see the different spatial topographies of these different systems across the human brain. So our lab and a big line of our research uses these neurochemical atlases alongside dynamic measures of brain signaling to try to understand healthy aging and then understand, probably more importantly, why people are starting to deviate away from healthy patterns of aging, healthy trajectories. The idea here is, first of all, we want to better explain why people are varying and how they're presenting symptomatically. So even though there's kind of these classical symptomatic patterns that we expect in Alzheimer's disease, we expect memory and attention impairments. In Parkinson's disease, we expect movement impairments that are kind of well profiled, as well as some more mild cognitive concerns. But people vary a lot in how they actually present in clinic with these issues. And it's really hard to predict with our sort of classical imaging approaches. It's hard to predict who's going to show more of a cognitive phenotype in Parkinson's, for instance, versus who's not. As a kind of step beyond that, we're hoping that these non-invasive measures could at some point be used for low-risk, repeatable biomarkers of these trajectories, right? Can we actually measure who's going to show one phenotype versus another and adapt treatment in a personalized way based on what we're seeing in that person's brain specifically? And then as more of a pie in the sky type of idea that we're really still hopeful for, from this, we may be able to also guide emerging pharmacotherapies, because in these disorders, oftentimes there's a lot of variability in how people respond to acetylcholinergic drugs or dopaminergic therapies. And we think that part of the reason for that might be because those drugs are hitting people's brains differently, and different people are having problems in neurochemically specific parts of the brain that may require different neurochemical targeting with these types of therapies. So I thought to structure the talk today that I'd kind of split it up into 3 pieces and not cover everything we've done in this area because there's probably too much to fit into one talk, but instead give a few examples of kind of interesting use cases where we can use this topographical analysis approach for brain imaging data to get at different questions and sort of at different stages of the research pipeline, contextualize our findings in interesting ways. The first of these is what's been conceptualized by Bratislav and Justine and their group as kind of like an enrichment analysis. So, gene enrichment, you basically do a big gene panel, and then you contextualize what you're finding there against huge databases of genes. And you kind of see what patterns are falling out throughout several genes rather than just looking at individual genes, which are probably much less informative. So the idea here is very similar. You have a topographic brain map of where in the brain things are changing relative to some external contrast, and you kind of do a one-by-all comparison. What neurochemical systems is this change mapping onto most strongly, or what set of neurochemical systems? The way that we do this in my group is a bit different from some other folks. We tend to use what's called a nested linear mixed effects modeling approach for this. I'm not going to dive into the nitty-gritty of why we think that's useful, but I'll kind of walk through the general pathway here before we dive in. Basically, we start with MEG data. We do all the kind of boring standard pre-processing of the data, make sure it's very clean. We get it to the level of the cortical surface, like what you see here, and we decompose the signal into those different brain rhythms, right? So we look at rhythmic patterns of fast, typically very fast brain signaling in these studies. We then take each person's brain map, each patient's brain map, I should say, who has the neurological disorder, and we standardize typically to the healthy control group. So we get, instead of a brain map of how this person's brain signaling looks on average, we get a brain map of how their signaling is changing relative to healthy levels. So this is like a Z-score map of the actual spatial pattern of change across that specific patient's brain surface. And then what we do is we feed all of that into a linear mixed effects modeling where we're looking at alignment of those brain signaling changes that are, again, sort of changes relative to healthy normal. And we do this for each person relative to these neurochemical atlases. So basically, if we have a neurochemical change relative to healthy levels on the y-axis here, let's say it's alpha changes relative to healthy controls in a patient with Alzheimer's disease. And then on the x-axis, maybe we have an acetylcholine atlas, right? Where higher values mean there's more density of acetylcholine on average in that brain region in most people, and lower values mean there's lower density of that system. And what we do is within this nested model, we fit a slope and an intercept for each person across brain regions. So each of these dots is going to be one brain region across the cortex. And we fit a model that basically tells us, is there a consistent trend in that person's brain where regions that are higher in acetylcholine also tend to be showing this change in brain signaling relative to healthy levels, right? So are we seeing changes in brain signaling in acetylcholine-rich regions for that person, right? We do that for each patient individually within this nested model, and we feed it into this larger linear model, controlling for all our error terms properly and all that fun stuff. And essentially what we end up getting out of that is a model where we can test those slopes, these relationships that we're modeling per person, against a null of 0, right? We can say, is there a consistent relationship across people where acetylcholine-rich regions are biased towards showing this signaling change in patients with Alzheimer's disease? But I think much more interestingly, what we can also do with this is we can look at interactions. So we can look at how these relationships are changing in people who have worse versus better cognitive function. Or we can say, are people with Alzheimer's versus people with earlier stage MCI and Alzheimer's disease showing a different relationship here, with the idea that from that you can get an indication of, for instance, maybe people who have more of these changes in acetylcholine-rich regions are the ones who are showing worse cognitive symptoms, and tying that back to sort of clinical variability in what we think is a really meaningful way. So when we've done this kind of analysis in a few different groups, we found kind of interesting and in some kinds really enlightening results. The first set of findings I'll show here are from the Quebec Parkinson Network. So this is a big database of patients, I should say, who are on dopamine medications who have Parkinson's disease. And what we saw in this study was that brain signaling changes that traditionally have been thought to be entirely pathological or entirely harmful in these patients, just because they're occurring in patients, so they must be problematic, right? Are actually aligned pretty strongly with nicotinic acetylcholinergic system densities across the brain. But importantly, what we saw that was really interesting and very new is that the more that these patients showed brain signaling changes relative to healthy levels in acetylcholine-rich brain regions, the better they were doing cognitively. So they were actually showing less cognitive problems if they had more of these changes in acetylcholine-rich parts of the brain. So that reconceptualized some of the changes that we've been traditionally studying as problematic in Parkinson's as potentially compensatory, right? If you're able to recruit brain regions that are rich in acetylcholine, maybe you're doing a bit better cognitively than people who have the same disease and have the same disease severity, but are not able to recruit those brain regions. We saw something similar when we looked at a different pattern of neurochemistry. So when we looked at mu-opioid and cannabinoid receptors, this one gets a bit complicated with double negatives in the explanation, but basically what we found here was that brain signaling changes in Parkinson's tend to avoid regions that are rich in mu-opioid and cannabinoid regions on average, but the patients who were able to recruit those regions slightly or a bit more than their counterparts experienced less psychiatric symptoms, which makes a lot of sense if you know about opioid and cannabinoid receptors and mental health. That's kind of an intuitive relationship. So in both cases here, we saw that these changes in brain signaling in patients with Parkinson's were potentially compensatory. We're actually helping people avoid psychiatric symptoms or cognitive symptoms. But depending on what they were aligning to neurochemically in the brain, we could actually predict which clinical symptoms they were going to be showing relative to their peers. Now, when we ran a similar analysis in patients with Alzheimer's disease, this is from the DMAP study that I led during my PhD and kind of a secondary analysis of the data that we collected there, we found, at least at the surface level, a similar set of results where we saw an alignment between brain signaling changes in patients with clinically defined Alzheimer's. So this is clinical dementia and mild cognitive impairment patients. We saw that there was stronger brain signaling changes in regions that are high in muscarinic acetylcholine receptors. So still acetylcholine, but a different subset of receptors with different dynamics. But importantly, what we saw here was that the folks who had more of these brain signaling changes in acetylcholine-rich regions were actually doing worse on tests of attention function, which is contrasting, obviously, with what we saw in Parkinson's disease. So in this case, this does seem to be more of a pathological relationship. It's relating to worse cross-sectional clinical function across individuals. And kind of supporting that idea, in this study, we also collected whole brain quantitative amyloid PET data. So we had measures for each person of where this harmful protein, that is what we think of as probably being at least somewhat causative in the disease process, we had that for every single person's brain. And basically, we saw that change in brain signaling in regions that are rich in muscarinic acetylcholine receptors seems to be entirely accounted for by how much amyloid is accumulating that brain region across people. So again, indicating this potential pathological pathway there. And then finally, to extend this a little bit more, we pulled in data from that PREVENT-AD cohort that I talked about before. This is a cohort where we have asymptomatic adults who, by definition and by the inclusion criteria of the study, do not have any clinical cognitive symptoms when they get enrolled. But some of them do accumulate amyloid and tau in their brains over the course of the study. They measure that with a quantitative PET through the course of the study. And some of them do progress to mild cognitive impairment throughout the course of this as well. And what we saw was, particularly in the case of these fast patterns of brain signaling, we were able to replicate those neurochemical relationships in this very early disease stage when people are still asymptomatic. So we contrasted people who have mild accumulation of amyloid that's getting to the threshold of being clinically significant against people who showed no amyloid accumulation. And we saw that the brain signaling changes in those amyloid, sort of marginally amyloid positive folks were matching onto the same neurochemical systems as what we showed in the clinical disease stage, and particularly with a really strong alignment with those fast frequency muscarinic acetylcholine alignments as well. So this looks, again, like it's tracking into the asymptomatic, very early sort of preclinical stages of the disease process. Great, so we can take a brain map and we can sort of throw everything but the kitchen sink at it, right? We can look at what it aligns with across all neurochemical systems, but oftentimes that's not really in line with what we're trying to do in a study, right? Oftentimes that kind of requires that the entire study is framed around this 1 by n sort of enrichment analysis approach. What if instead we have an interesting relationship in our data that we think might be specific to a neurochemical system, and we want to provide or not provide converging evidence for that neurochemical specificity? What do we do now? A good example of this came up in a recent study that we put out last year, or I guess this year with the new publication date. where we were looking at degeneration of deep brain nuclei in patients with Parkinson's disease. So in Parkinson's disease, there's kind of two parts of the brain that degenerate very early and very preferentially in this disease, and these are in the brainstem. So we see degeneration, death of cells in the substantia nigra pars compacta, which you can see highlighted by the hyperintensities on the images on the top left here, as well as the locus ceruleus, which are actually these tiny little dots on the bottom of the images on the top right. Very difficult to measure until recently with non-invasive methods. So here we're using a special sequence called neuromelanin-sensitive MRI that gets a really nice contrast in these that wasn't really available until the last few years. And basically, in terms of substantia nigra degeneration, this is a pathway that's very well studied in Parkinson's. Substantia nigra projects through the nigrostriatal pathway, coordinates movement, and it's kind of the primary basis of why people are struggling with movement in Parkinson's disease. There's death of cells in this region, leads to changes in signaling eventually at the level of the cortex, and that's related to the severity of motor symptoms. We found all that stuff in this study. That's not the point of this at all. But what we also looked at in the study, again, was this locus ceruleus structure, which is neurochemically specific. The locus ceruleus provides the vast majority of norepinephrine for signaling at the level of the cortex in the human brain. So basically all the norepinephrine you're using for signaling for things like alertness and cognitive control and all the important things it's used for is produced in these two tiny little white spots very deep in your brainstem. So we measured the integrity of this in patients with Parkinson's disease. We found, as we expected and based on previous literature, we saw decreased integrity of the locus ceruleus in patients who have Parkinson's disease. And then we took those measures of locus ceruleus, sort of deep brain nucleus integrity, and we regressed them on brain signaling using kind of a standard classical approach like what I was talking badly about on a previous slide. So we took our locus ceruleus measures, we regressed them on rhythmic patterns of brain activity across the entire cortex. We controlled for nuisance covariance, age, education, all that fun stuff. We also controlled for integrity of the substantia nigra with the idea that we're looking for nucleus-specific patterns here. We don't want to just look at general degeneration of one region or of the whole brain. We want to look at specific degeneration of different regions relative to another. And what we saw was this really interesting pattern that's a little bit washed out on the projector, unfortunately, where these frontomotor regions of the brain seem to be showing this really strong association where lost integrity of the locus ceruleus was related to pathological or at least Parkinson's disease looking increases in alpha frequency signaling and brain signaling at that location, right? We also saw that this was relevant for cognitive function. So basically this relationship was stronger in folks who were struggling more with their cognitive abilities at the time that these measurements were taken. And that's by itself, I think, pretty interesting. It's entirely correlational, unfortunately, but it does kind of hint that there might be this new neurochemically specific pathway where loss of cells in this neurochemically, this norepinephrine specific structure in the deep brain are related to changes in brain signaling at the level of the cortex with relevance for symptoms, for cognitive function. But of course, that's, again, the hand waving I don't like, right? We don't want to just wave our hands and say, since this is a neurochemically specific nucleus and we're seeing it correlated to brain activity changes, it must all be this neurochemically specific pathway. We wanted to shore that up a bit and hopefully look for converging evidence from a different method. And that's where this neuromaps approach really came in handy and was really useful here. What we did is we took this map that you see here and we unthresholded it because we like unthresholded maps now. We recomputed it at the sort of the spatial level that's useful for being able to compare to the atlases that we have, just to make things statistically comparable spatially. And we looked at whether the changes in brain signaling that we saw related to degeneration of this norepinephrine specific nucleus were occurring preferentially in regions where we expect sensitivity to norepinephrine. That's intuitive, right? If this is a neurochemically specific effect, then we expect it to be in the regions where we expect neurochemistry around that system, right? And what we saw was exactly that brain regions where we expect lots of sensitivity to norepinephrine that have a high density of norepinephrine transporters, those were the regions where we saw that negative going relationship between degeneration of this norepinephrine specific nucleus and brain signaling changes in patients with Parkinson's. So in this way, we were able to provide converging evidence that maybe this effect is really neurochemically specific. It's obviously not a perfect causal pathway. It's nowhere near that because what we do with imaging is typically inherently somewhat correlational. But to me, at least, this really shored up the idea this might be a neurochemically specific effect and that this pathway might be really a bit more substantive than just a simple correlation between two measures. So the last thing I wanted to touch on for ways that we can use these methods, and I think interesting ways, is a new approach that we've developed to, instead of sort of taking a brain map you've already computed and co-localizing that with neurochemistry and seeing what falls out and seeing if it is an interesting spatial pattern essentially in terms of neurochemistry, we can also actually take these neurochemical atlases and we can use the spatial pattern in that atlas to search the brain data that we're interested in. So instead of taking statistics on the back end and then using the neurochemical maps to contextualize that, we can actually use these on the front end to derive our statistics in the first place. And the way that we can do this is using a partial least squares framework, like what I talked about earlier. I'll dive into that in a second. Basically, for this study, the impetus behind this is that patients with Parkinson's disease essentially always take dopamine replacement medications. I touched on the substantia nigra earlier and how that's really important for movement in patients with Parkinson's. That system is, or at least the degeneration of that system is entirely based around dopamine function, right? So if there's Parkinson's people in the crowd, and I've been talking up here at all of you, and haven't said a word about dopamine, you might be wondering why that is. Like, where's dopamine in the Parkinson's story if this is a Parkinson's talk partially? And the answer to that is that all of the patients in these studies have been so far on their dopamine medications. So all of the changes in brain activity that we'd expect due to degeneration of dopamine systems are likely normalized at this point because they're taking medications to normalize that effect. And the other thing that I think is really important to point out is that the dopamine systems that get affected by Parkinson's are not typically thought to be cortical systems, right? So we see degeneration of the substantia nigra, which then leads to problematic signaling throughout the nigrostriatal tract. That all occurs before anything hits the cortex. And by the time we're at the level of the cortex, dopamine systems are not what's being used to signal in the regions where those effects are taking place, right? But in line with that, there's kind of this persistent issue in the field where, since every single patient is taking these dopamine medications, we're not giving them specific dopamine medications, right? They're not targeted only towards the brain regions where we hope to normalize signaling. We're actually flooding their entire brain with dopamine and hoping it hits the right region, along with some other regions that we're potentially not hoping to hit. So because of that, there's been this question in the field over how much off-target effects are there of these dopamine drugs on things like cortical dopamine systems, which there are dopamine receptors throughout the cortex, but they're not what we're hoping to target with these levodopa and dopamine replacement therapy drugs, right? So what we wanted to study here was these off-target effects. We wanted to say, if we're not trying to hit the cortex with dopamine when we give these therapies, if we do hit the cortex, what happens? And that's a really difficult question to answer, because how do you know where in the cortex dopamine-specific activity is happening, right, when we give people these medications? So what we did here is we had a study with collaborators from Karolinska, where they had MEG data from patients who were both on and off their dopamine medications. So they had them come in off their medication, they did a scan, they had them take their medication in the center, and then they redid the scan once they were on their medications and stable. And from that, we get these neurophysiology maps really similar to what we had before, but we have these, again, both on and off medication. We can then take the difference of these and see what the effect of medication is on the brain, right? On the cortex, at least, of these dopamine medications, which, again, we're hoping not to hit the cortex too much with, because that is likely not going to be too helpful. We then do something similar where we standardize these dopamine effect maps to similar maps from the controls. Obviously, the control participants don't take the drug, but they do a pre-post session that's equally timed to make sure everything's not just due to sort of confounds in the data collection. And we get these maps that tell us where dopamine is having an effect on the brain or the brain signaling in each of our patients with Parkinson's. Again, what we can then do with that data is we can use our neurochemical atlases in a partial least squares approach to actually search those maps and find where in the brain there's systematic but variable changes in neurophysiology as a result of that medication in these brain regions that are really sensitive to dopamine. So basically what we're saying is, can we find a spatial pattern based on these atlases of where we expect potential off-target effects of dopamine therapies on the brain signaling maps of these patients with Parkinson's disease? From that, we get these what we call latent components, which are basically maps of where we see that alignment, where we see brain regions where we don't want to be hitting being activated by these dopamine therapies in terms of their signaling. And really importantly, we get a weight per each participant. So we get actually a measure that tells us how much each patient is showing that off-target activation of these cortical dopamine systems by the medication that they're taking. So when we ran this in what is admittedly a relatively small sample of folks, it's really difficult to collect these on-off medication data in folks with Parkinson's. We found this really strong sort of systematic spatial pattern where these inferior frontal and temporal regions that we know are really dense in dopamine receptors in the cortex are being activated variably across patients by these dopamine therapies. You can kind of see, if we plot this kind of per person with some example brains, you can kind of see what we mean by this, where some folks, like the patient up here, so this is just one patient's dopamine response map, are showing a really strong activation of these sort of dopamine-rich regions of the cortex, whereas other patients are essentially showing a flat map, where they're not showing any meaningful activation of these dopamine-sensitive parts of the cortex by the dopamine therapy. And much more importantly, what we saw was that the degree of off-target activation of these dopamine-rich parts of the cortex was explaining who was responding better to the therapy. So the folks who had more of these potentially problematic off-target cortical activations were doing less well when they took their medication versus the people who had no such effect and were doing much better. Great, so that's probably a lot. That's kind of the end of the neurodegeneration, age-related neurological change part of the talk. But I thought I'd reserve the last couple minutes of the lecture to go into like where we're going with this and what's kind of new in our thinking process in this area. And probably the most interesting thing that we're doing in this sort of research landscape currently is being led by Santiago Isaac Flores Alonso, who's a really talented postdoc in the lab, has been here for about a year. And what he's been asking is instead of, can we contextualize these single region changes or these topographies region by region with these neurochemical atlases? He's instead interested in, does neurochemistry shape the regional connections between parts of the brain, right? So again, we know that the brain does not process information in isolation. With these neuroimaging measures, we can easily compute statistical associations between the signals in different brain regions and talk about how different brain regions seem to be processing similar information if their signals are very similar. And what Santiago's been asking is, if we do that connectivity mapping, right? If we say these two regions are processing things more similarly than these other regions at different parts of the brain, does that align with how similar those brain regions are neurochemically? So what we can do is we can take our neurochemical atlases, we can region by region take the profile, like you see here, of each region of the brain in terms of its neurochemistry, and we can do a big correlation matrix, right? We can say these two brain regions that are in red here are showing a very similar neurochemical profile. They have high levels of norepinephrine and low levels of serotonin and et cetera, whereas ones in blue show very different neurochemical profiles from one another. And what he's been seeing is that, kind of as a spoiler and from our early findings, the connectivity, in this case, we're showing the beta band connectivity on the x-axis here, does seem to be facilitated or shaped somewhat by how similar two brain regions are in terms of the neurochemistry. So regions with more neurochemical similarity that show a more similar pattern of their brain chemistry are showing stronger connectivity than regions that are not. So this is kind of showing the same effect, but across different frequency bands. This is probably more interesting for some of the MEG, EEG nerds in the audience, if there are any. But basically, we're seeing this is somewhat specific to that really high frequency beta band, but it is significant across different frequency bands, especially the more rhythmic bands like alpha and theta as well. The only one that's not significant is in the high gamma band. And using a leave one out approach, what we can also do is ask, which neurochemical systems seem to be playing into this the most? And we saw some interesting specificity here where in the high frequency beta band where we're seeing the strongest effects, serotonin seems to be really important for shaping long distance connectivity, whereas in these slightly lower frequency rhythmic bands in the theta and alpha range, we're seeing that norepinephrine is playing an outsized role. It's really important in shaping interregional connectivity between brain regions. We also, and I think really relevant for this talk, we're seeing that this is being shaped or being changed by age and by cognitive function. So we're seeing that folks who are older tend to have more strong of an influence on neurochemistry on brain connectivity. And I think maybe even more interestingly, when you control for age and other confounds, we actually see that particularly in the beta band, folks who have more of this neurochemical orchestration of long distance brain connectivity are also doing better on tests of things like fluid attention, which I think is really interesting and indicates that this might actually be relevant for external behavioral characteristics and cognitive function as well. So from there, where we're looking to go next, we want to see how well these neurochemical connectivity alignments do in terms of explaining symptoms. Of course, we're not interested here in just sort of staying in the aging and healthy aging domain. We want to see if this actually is changed by age-related pathologies. And more importantly, does it help us explain why people are doing better or worse in these pathologies? And more importantly than that, does it actually give us anything new? So we can actually benchmark prediction of, let's say, clinical group or clinical symptom severity from these regional metrics that I showed earlier in the talk against our connectivity metrics and see if they're actually adding anything new or if it's all just based in the region-wise information or not. Kind of counterintuitively, we also want to work backwards. So all of the work that I showed today is largely kind of confined to the really clinical symptom stage of neurological disease. So we're talking patients who have Parkinson's and are in clinic already, people with dementia or cognitive impairment. We want to make sure that this is actually useful as an early stage biomarker. So the next step there is to take this back several steps and look at whether these relationships are identifiable in what are considered essentially healthy adults who have higher retrospective risk for age-related neurodegenerative disorders. And then finally, again, kind of counterintuitively, the big thing we really want to do is scale this down, not in terms of sample size. We always want lots of people in our studies. But what we want to do is take MEG measures, which are admittedly relatively inaccessible and expensive to use, and see if we can find which features from the MEG signal are actually extendable and measurable with much more accessible and cost-effective technologies. Things like EEG or potentially optically pumped magnetometry, which is a new method of MEG that hopefully will really reduce the cost of MEG and increase the accessibility soon. But the key thing here is we really want to make sure that this is actually useful in a much broader sense. So we want to scale these to more accessible technologies as well. So with that, need to thank a whole lot of people for this. For the most recent stuff I showed, especially Santiago, who's here, and the rest of my crew here at SFU. A lot of the work that I showed was through external collaborations that I've been running through my postdoc and into my faculty position, or in some cases in my postdoctoral work itself. So huge thanks to everyone who is a mentor or a collaborator on the work that I showed here today. Of course, have to thank our many funding sources, and then a big shout out to the open data repositories that also supported this work. So PreventAD, Omega, QPN, and CamCan especially are these giant data repositories where you can look at all the data that I was showing here today and do your own fun, fancy things with them with relatively minimal effort. I should say minimal effort to get the data, not to process it. That takes a lot. But these are these really fantastic resources that need to be, I think, given better recognition for the work that they've put in. So yeah, with that, happy to take any questions, and thanks for listening.
Hey, thank you so much, Alex, for a really great talk. And thanks for everyone's contributions with great questions.
Yeah, thanks everyone for coming.